in the genetic segment, understand that while we talked about cancers being systematically associated with modifications that happen in the genome, the question comes, what specifically is happening in the genome? What kind of modifications are we talking about? So there are two major types of modifications I want you to remember. One, it's what we call the driver mutations. The driver mutations are these mutations that when they occur, they are very likely when expressing or not expressing to lead to a cancer, right? These are driver mutations. These are the one that very likely are going to lead to cancer when a modification occurs, right? So here we have, for instance, the proto-oncogenes that get modified to overexpress, and at this point we call them oncogenes, right? So these are going to basically lead to a cancer situation. The second group of genes that I already mentioned are the tumor suppressor genes, which can lose their function and therefore unable to stop cell division, right? Also, the last one, it's going to be chromosome rearrangements, right? This chromosome rearrangement can also lead to cancer. These are some examples of driver mutations. Now, so far, there are about 200 genes that are known to be driver mutations. Now, other mutations are going to occur. These mutations are called passenger mutations. These passenger mutations have these characteristics that they do not necessarily lead to cancer, right? These mutations occur, I want to say, almost all the time, if not more often than driver mutations. These mutations occur in germline cells, they occur in somatic cells, they occur relatively all the time throughout our lifespan. However, these mutations never really lead to a cancer situation. They don't trigger cancer. They don't encourage cancer. They don't cause a cancer to go, for instance, from benign to malignant, right? To metastasis, right? So they don't typically cause that. So these are mutations that are going to be called passenger mutations because they are just there and they occur and they are not drivers. Just as simple as that, right? So these mutations occur both in cancer cells and non-cancer cells, regardless, right? We record thousands of genes that actually carry these mutations. Now, when it comes to the mutations, whether they are happening in driver genes, we are going to record what we call a three strike. Three strikes because you are going to have a cascade of events of mutations. Well, it's not that it's set in stone. It's just that, well, you have one first event is going to happen. Then there is another event that needs to happen for the cell to progress toward a cancer, and then there is a third event. Now, let's dive into these different events. So, the first event is what we call a breakthrough. A breakthrough, it's basically when a first mutation occurs in a driver mutation. A driver gene, right? That is going to be a driver mutation. It can be a proto-oncogene, an oncogene, or even a DNA repair gene, right? So that's a breakthrough, right? So here we have a single cell that is going to be mutated, that is going to start losing its normal behavior, right? It's going to start moving toward uncontrolled cell division. They somewhat undergo uncontrolled cell division, but it's not bad yet. It's not bad yet. So then you have a second strike. That second strike, it's what we call expansion. That expansion is going to happen in a driver, as a driver mutation. And that is going to cause the cells now to multiply much faster than they were multiplying in the breakthrough. Okay, now understand that a breakthrough has to first of all happen, right? So the cell is kind of like dividing already fast, but with expansion, it's even faster, right? And while this are multiplying faster, you are going to, this mass of cell is going to be in a condition where there is need of nutrients, need of oxygen, need of energy, but that oxygen, nutrients, and energy are not flowing as fast as the cells are multiplying. Therefore, these cells have to be in a physiological state that allows them to withstand somewhat a situation of lack of oxygen and lack of nutrients and the inability to get rid of waste. That is the very reason why there is a need for angiogenesis to occur at this point. And angiogenesis, as a reminder, it's the growth of new blood vessels. Not just only new blood vessels are going to grow, but we are also going to have lymphatic vessels that are going to grow in these areas, right? So that is the rationale for the need for angiogenesis in expansion stage. And then you have a third strike. That third strike, it's the invasion stage. So there is another modification that is now going to lead to a situation where cells not only they lose their normal function, they multiply way faster than they should, but in addition, they are going to lose the ability to actually attach to each other. By losing that ability to attach to each other, well, needless to say that they're going to break away from the tumor and spread through the body, right? So, and this is when we going to name the stage where the cells break away and go in circulation, go multiply in other places, and spread throughout the body, metastasis, right? So we have here some cells getting away from the primary area where the tumor developed and going in circulation and spreading in other places of the body. So we have some extra blood vessels that are developing to actually be able to sustain the need in terms of nutrients and oxygen and getting rid of waste of this tumor, right? So this three strikes is very important to remember. So breakthrough, expansion, and invasion. Now, the three strikes have been identified and associated with many cancers as they go from initial driver mutation to evasion driver mutation. Now, some cancers, including melanomas, pancreatic cancers, cervical cancers, and colorectal cancers, have been well characterized. Now, if we take, for instance, the case of melanomas, BRAF, it's a gene that has been associated with breakthrough. This is a driver mutation. Then we have TERT gene in melanomas that has been associated with expansion, and another set of genes associated with invasion, including CDK2NA, P53, TP53, and PIK3CA. Now, for pancreatic cancer, for breakthrough, we have KRAS. Then for expansion, we have CDK2NA, and for invasion, SMAD4 and TP53. Now notice that some of the genes here are associated not just with one cancer, but with multiple cancers. For instance, TP53, okay, CDK2NA are associated with melanomas and pancreatic cancer. And as we look at many other cancers, we see that some genes in which there are driver mutations are involved in many cancers, right? So I'm not going to walk you through all the cancers here, but you can see for yourself that for cervical cancer, for instance, we have TP53, okay, that we already mentioned for previous cancers, and colorectal cancers, TP53 here again, right? KRAS that we are mentioning here in colorectal cancer for expansion, and KRAS for pancreatic cancer is going to be for breakthrough. So you see that, well, for a particular gene that undergo mutation, that mutation can represent a driver for one type of cancer, an invasion for another type of cancer, an expansion for another type of cancer. It just depends, right? Now, let's take a look at the evolution of cancers. Now, the evolution can be linear or it can be branched. What do we mean by that? Well, in a linear evolution, you have a parent cell here that is normal. That cell somehow gets mutated. That mutation can be caused by some environmental factors, mutagens, radiations, chemicals, or it can be intrinsic because of lack of integrity in copying DNA. Either way, there is a mutation, right? And then, following that first mutation, that cell, as it multiplies, the same cell, right, kind of cell, is going to receive another mutation. So that second mutation is going to take the cell toward more and more toward cancer or different level of multiplication of the cell. And then from that mutation, that second mutation, a third mutation is going to occur, right? So now we're going from 1, 2, 3 mutation. So 1, 2, 3 mutation. And then some other mutations can occur, and these mutations are going to cause these cells to change their characteristics as they accumulate the mutations. This is the case of linear mutations. And then we can have branched mutations, right? In branch mutation, we have a normal cell here that receives a strike here, and that strike leads to a first mutation. Now, from there, that cell can receive another mutation, another strike for another mutation, or another strike for a mutation that is going to happen at a different location, so that we have now this cell gets a strike here, right? And that same cell gets a strike now right here, so that you have that cell now that is now a kind of different kind of cell. But in this group of cell, another strike can happen at another location, so that now we have a variant of this cell 2 times. So we have 1, and we have 2. And now each of them is going to go their own way of evolution, including another strike that can happen here and another strike that can happen here. Okay? So that from this cell number 1, now we have another 1 prime and 1 second, which are different cells. But now let's take a look at cell number 2 here. Now that cell number 2 itself is going to undergo some mutations, including here, and more of mutation that is kind of linear here. So really, what we see here in a branched evolution, it's that at the end of the day, you are not going to have one single and exclusive type of cancer cell. You are going to have multiple cancer cells, right? So you're going to have this cancer cell, this cancer cell, this cancer cell, this one, so that, well, all cancer cells may not be exactly the same genetically. You have that variation of genetic background of different cells here. So cancer evolves in a person just like evolution occurs among living organisms, right? So seeing some drastic changes from what we call the parent and the offsprings. See a difference that is happening, right? From ancestors to descent, right? We can see these differences, right? So, and this is how, by looking at the small differences among organisms, we can say that, okay, well, while these two organisms are closer together in terms of evolution, nevertheless, or they are distant in terms of evolution, nevertheless, they are associated to a common ancestor who is this organism. I'm not going to go into the specifics here, but what I want to say is that when we look at a cancer that is metastasizing in the body, right, we see that we have different types of cells that are cancer cells in that individual. However, all these cancer cells can be traced back to one original cancer cell that is going to be this cancer cell right here. All right? So, now, let's take a closer look at what is happening at genes that are involved in cancer cells. Now, again, we have proto-oncogenes, tumor suppressor genes, and DNA repair genes. Let's take a look at specifically now proto-oncogenes. So proto-oncogenes, as you already know, they promote cell division. They act in our cells like a gas pedal, right? You hit the gas pedal, the car starts running, okay? So its expression has to be tightly regulated. An overexpression, it's a problem and leads to cancer, right? So when it comes to proto-oncogenes, for them to be associated with cancer, they have to gain a function, right? And that gain of function can be triggered by a point mutation, chromosomal translocation, or inversion. All these changes can cause an overexpression of a proto-oncogene, and at that point, it's not a proto-oncogene anymore, it's an oncogene, right? So we have a gain of function that is dominant. Then, other thing that we need to say about proto-oncogenes, so they can also become oncogenes by being physically positioned close to an area that expresses all the time. Okay? So in a case of a translocation, right, or the movement of one gene from one place to another, where, well, whenever that DNA moves from one place to another, the proto-oncogene can accidentally find itself close to an area that is highly transcribed. For instance, genes that are responsible for the production of antibodies are expressed all the time. So, if a proto-oncogene moves closer to that kind of gene, needless to say that proto-oncogene is now going to become an oncogene because the expression is going to follow the pattern of expression of the antibody gene. All right? So, the situation that can cause this kind of overexpression by physically locating the proto-oncogene in an area of high gene expression, it's for instance in the case of viral infection, right? So when a virus comes, inject its genome, well, its genome goes and insert about anywhere, and that anywhere can be close to a proto-oncogene, right? So at that point, the viral genes are going to drive the expression of the proto-oncogene that is now going to become an oncogene, right? Another example, it's when the proto-oncogene is moved close to a gene that is encoding a hormone. Now, hormones are produced all the time in our body, right? And remember that hormones are very heavily involved in homeostasis. And genes that are involved in homeostasis have to be expressed all the time for the single reason that the body is trying to reach a balance all the time, maintain a balance all the time. Not sometimes, but all the time, right? Which explains why when there is such movement of a proto-oncogene close to a gene that encodes a hormone, it's going to be overexpressed, right? Or, I already mentioned this, a position of the gene close to an antibody gene. All right. So we have some viruses, including the Epstein-Barr virus, hepatitis virus, herpes virus, HIV virus, right? And HPV, human papillomavirus. They have all been associated with cancers. For instance, the Epstein-Barr virus have been associated with lymphoma, all right, with stomach cancer, and the throat cancer. Hepatitis C virus has been associated with virus of the liver. That is the reason why, by the way, it is called hepatitis. Okay, hepatitis C. I'm sorry, not hepatitis B. Hepatitis C virus, right? Herpes virus 8 has been associated with a type of cancer that is called the Kaposi sarcoma. Okay. HIV has been associated with many cancers, including the Kaposi sarcoma, the cervical cancer, the B-cell lymphoma. HPV has been associated with the cervical cancer, throat cancer, anus cancer, vaginal cancer, and penis cancer. Okay, so this is just to show you how much viruses are associated with cancer. And again, when this virus comes in, they bring their viral particles and they insert about anywhere. When I say viral particles, I'm really talking about the genetic information, right, that insert in our genome and cause overexpression of a proto-oncogene. All right. So now, because of what I already mentioned on overexpression and because of the amount of material, I'm not going to dive into the details here that really I scheduled to talk about, but I'm not going to talk about them. So let's move on here. So just know that, proto-oncogenes are going to overexpress, right? That's what leads to cancer, okay? All right, so here we have the case of the Epstein-Barr virus, right? That actually is going to cause the Burkitt lymphoma. And that Burkitt lymphoma is actually happening because of B cells, right? And the characteristic of that lymphoma, it's a deformation of the jaw, right? And you can see how the jaw is deformed in this child, okay? Now, a change in the translocation. So here we have a graphic representation of what this means. So here we have chromosome 8 and chromosome 14. And on chromosome 8, we're going to have the proto-oncogene that is located in this area, right? Now, the reason why you see two chromosomes here, it's because the DNA has been copied, right? The cell has gone through the S phase. Now we have a centromer. We have one sister chromatid and another sister chromatid, okay? You understand that. So now, that proto-oncogene is located on chromosome 8. Now, on chromosome 14, we have a gene encoding antibody, right? That gene is located right here, all right? So now, in a translocation that occurs between chromosome 8 and chromosome 14, we have a translocation such that, well, this segment is going to move here, while this segment is going to move here. As consequence of that, we have a translocation here and here. Now, what does that mean in terms of cancer? Well, this means in terms of cancer, it's that now this proto-oncogene right here, okay, is moving now close to this antibody gene. By being close together now, well, the proto-oncogene is not a proto-oncogene anymore. It's an oncogene that is going to be expressed all the time, okay? So that is the case of translocation. Then we have the case of alteration of a chromosome so that an oncogene is going to move to a position that now depends, well, in a position in which the expression of the proto-oncogene depends on another gene, right? Causing the proto-oncogene, at least the open reading frame, right? The open reading frame of the proto-oncogene is now going to merge with the open reading frame of another gene, causing a hybrid protein to be produced. That hybrid protein, it's actually what we call a fusion protein. So that protein is going to be produced out of one single transcript, right? That transcript is going to contain the open reading frame for the gene, right? And the open reading frame for the proto-oncogene, which at that point is now an oncogene. So the two proteins are going to form one single big protein, right? This is what we call a protein fusion, right? So we see this, for instance, in acute promyelinic promyelicytic leukemia, okay? So this is the case of a translocation. It has to be a translocation, right? It has to be a translocation because for the proto-oncogene to move from one location to another, that has to be a translocation, right? Now, translocation, in this particular case, it's happening between chromosome 15 and chromosome 17, right? I'm not going to bug you with the details. All right. So then we have another type of cancer, the chronic myelogenous leukemia. The chronic myelogenous leukemia. In the case of the chronic myelogenous leukemia, this is going to happen as a translocation that occur between chromosome number 9 and chromosome 22. So here, two genes, the gene ABL on chromosome number 9 and the gene BCR on chromosome number 22, they are going to come and merge together in a way that they are going to produce a fusion protein that forms the protein BCR-ABL, right? That BCR-ABL, it's actually an oncoprotein. An oncoprotein that is a tyrosine kinase. Now, a tyrosine kinase is particularly important to mention in the case of cell communication, what we call transduction signal pathway, right? That transduction signal pathway can be oftentimes amplified. And that amplification is going to unfortunately mean overexpression, right? And cell turning into cancer cell, right? So here, obviously, because of that translocation, we are going to have the development of a cancer. Then we have the case of growth factor receptors. Now, growth factors are these molecules that trigger the growth of cells, tissues, organs, right? Here we are going to spend some time on HRE2. HRE2, it's actually a growth factor receptor, right? Now that growth factor receptor, okay, is going to bind a growth factor. Now, if the growth factor receptor is modified such that it has the impression that a growth factor is present there all the time, that growth factor receptor is going to be activated all the time. Or if the growth receptor becomes hypersensitive, well, then that growth factor receptor is going to be stimulated all the time. And if it is stimulated all the time, this is going to cause cancer, right? So epidermal growth factors are typically these factors that are tyrosine kinase receptors, right? Tyrosine kinase mean that these receptors are not just simply receptors, they are also enzymes, right? They are also enzymes that are involved in cell communication and specifically in the transduction pathway that most of the time is going to lead to the multiplication of the cell, right? So now, oftentimes, when these receptors are hyperactivated, hyperstimulated, this leads to cancer, right? So now we know that there is a monoclonal antibody-based drug that can be used to attach to on these receptors so that they block the hyperactivity of the receptor.